cellular part of biochemistry. So in the cellular part, first we need to start with cell cycle. So cell cycle is divided into interphase and M phase. In interphase you have again G1, S phase, G2 phase and M phase. And in M phase there is a prophase, pro-metaphase, metaphase, anaphase and telophase. So this is a basic part we know. And after this all there is a cytokinesis process. You can see where the cells are divided into two daughter cells. So this process is mitosis. So before mitosis, whatever goes on is called interphase. So in this phase, the cell prepares itself to undergo mitosis process. So there are several checkpoints like G1 to S. Between G1 to S phase there is G1 to S checkpoint, and between G2 to M phase there is one more checkpoint, and even before cytokinesis, that is between M phase and cytokinesis, another checkpoints. So all these checkpoints are in turn regulated by cyclin dependent kinases. You can see in this diagram clearly cyclin dependent kinases. But this cyclin dependent kinases alone can't do this. It is activated by cyclin. And this cyclin dependent kinases complex is in turn activated by mitogens. You can see here mitogens are nothing but growth factor which stimulate the cell division via CDK and cyclin complex, especially in the S phase of the cell cycle. In the S phase of the cell cycle only, the cell cycle gets activated whenever it needs to get activated. Remember, in the S phase only. So this CDK and cyclin complex, in the name itself you can see CDK, K means kinases. These are kinase enzymes, cyclin dependent kinases. So the kinase enzyme always phosphorylates the protein. Here this is a retinoblastin protein which is phosphorylated. As it is phosphorylated, it releases a transcription factor which activates the S cycle so that this cycle continues. So this is like an accelerator for the car. For example, you think this cell cycle system, especially this cancer related one, as a car which has an accelerator and a brake, and the accelerator is a cyclin dependent kinase and the brake is a tumor suppressor genes like P53. So whenever there is a problem in accelerator, like if you over accelerate the car, that will lead to accident. The same thing, if you over accelerate this cell cycle, then that will lead to cancer. At the same time, if the brake has failed for the car, that will also lead to accident. That is, if the tumor suppressor has failed, then you are not able to suppress the tumor, so that again the cell cycle will get overactivated and that will lead to cancer. So now we'll discuss about tumor suppressor. Tumor suppressor P53 activates P21 and this P21 will inhibit the CDK. So this is a tumor suppressor gene. So this will inhibit the CDK. So because of inhibition, the cell cycle will inhibit. But if this get mutated in some diseases, then that will lead to cell cycle, because overproduction of cell cycle, and that will lead to different mutations. So especially there is a syndrome called Li-Fraumeni syndrome. In this syndrome there is a mutation in tumor suppressor gene, so that will lead to overproduction of the cell cycle. So because of that you can get different kinds of cancers. The mnemonic is SBLA. That is, you can get sarcoma, breast cancer, leukemia, adrenal cancer. All these cancers are produced because of suppression of P53 tumor suppressor gene. So we have understood the cell cycle phases, checkpoints. We have also understood the regulatory CDK and tumor suppressor. Now we'll come to cell types. So cell types are of three types, that is permanent cell type, stable and labile. Permanent cells are like they are always in G0 phase. G0 phase, you can see here, this is a phase where the cells are inactive, like neuron cells, skeleton cells and cardiac muscle RBCs. They are always inactive. They are permanent cells. They don't again come into the cell cycle. Whereas stable or quiescent cells are which can enter again the G1 from the G0. So these cells are like hepatocytes. It can regenerate again if you start it, and lymphocytes, PCT, periosteal cells. All this can again come to G1. Next, there are labile cells which are never going to G0. They're always in this cell cycle. They're always continuing in this cell cycle. They divide rapidly with a short G1, and they are mostly affected by chemotherapy because they are always rapidly growing. So whenever you give chemotherapy drugs which actually inhibit this cell cycle, so this cycle will affect this kind of cells, that is bone marrow, because it is always rapidly continuing, so that will lead to myelosuppression in chemotherapy, and gut epithelial cell that will also decrease, and skin and hair. So that's why there's alopecia in cancer patients after giving chemotherapy, and also follicles and germ cells. All these are also affected when you give chemotherapy. So these are the cells which are always dividing. Now let us discuss about rough endoplasmic reticulum and smooth endoplasmic reticulum. So actually this endoplasmic reticulum is a site where you produce proteins and lipids, especially rough endoplasmic reticulum where ribosomes, you can see they're present on it, which produce proteins. Whereas smooth endoplasmic reticulum doesn't have any ribosomes, they, you can see smooth surface, so they directly produce lipids. And one more thing you need to know is in the neuron, the Nissl granules which are present, actually it's a rough endoplasmic reticulum for the neuron, right? So that will produce proteins within the neuron by Nissl bodies, okay? And sometimes there are some free ribosomes not only attached to this rough endoplasmic reticulum. For example, like this, these are the free ribosomes, and these are unattached to any membrane, right? So they synthesis mainly cytosolic proteins like peroxisomal proteins, mitochondrial, all those proteins are produced by this, which are outside the membrane like this in the cytoplasm. And one more thing you need to know about rough endoplasmic reticulum is there are N oligosaccharides. N oligosaccharides are especially present in rough endoplasmic reticulum. Okay, N, make it capital because N oligosaccharides are present in rough endoplasmic reticulum. And especially this rough endoplasmic reticulum is more abundant in mucus secreting goblet cells of small intestine because that is a protein, more protein is required. In antibody secreting plasma cells, which is also a protein, and also pancreatic beta cells, which is also protein. So wherever there is a requirement of more protein production, there will be more abundant of rough endoplasmic reticulum. And the proteins within the organelles, even for different organelles also you need proteins, like for Golgi, for lysosomes, you can see here this is a lysosome, this is a Golgi. So for Golgi and lysosome also proteins are required, and even those proteins are also formed from the rough endoplasmic reticulum. Now of course smooth endoplasmic reticulum, it produces lipids or steroid synthesis, and also it is having important function, that is detoxification of some drugs and poisons. And it also is the location for glucose 6 phosphate. Glucose 6 phosphate is present here, okay? Glucose 6 phosphate is present here, which is last step of glycogenolysis and gluconeogenesis. Hepatocytes, steroid hormone producing cells of adrenal cortex, gonads, all are rich in SCR, because in gonads, estrogen, progesterone, all those are lipids, and in hepatocytes you need some lipid products, LDL, like that. Steroid hormones from adrenal cortex, glucocorticoids, all those are produced from smooth endoplasmic reticulum. Now let us understand about this process, like how from the endoplasmic reticulum the products are moving to Golgi, and how it is able to make vesicles and pack them, and it is sending out to plasma membrane. Let us understand this. So actually Golgi makes O type of oligosaccharides. So N type of oligosaccharides from the rough endoplasmic reticulum come to Golgi, and they are converted into O type of oligosaccharides, especially in serine and threonine. You can see here clearly N type of oligosaccharides are present on asparagine, whereas O type of oligosaccharides are present on amino acids serine and threonine. And they also add mannose 6 phosphate. So mannose 6 phosphate. G6P is present in smooth endoplasmic reticulum. Mannose 6 phosphate is added in the Golgi apparatus to the proteins. Especially it is also added to lysosomes. And one more thing, Golgi is divided into cis side and trans side. You can see this is a cis Golgi, which is towards the endoplasmic reticulum. Trans Golgi is towards the plasma membrane and lysosome. And very important function of lysosome is the one which triggers the protein package on the trans side of the Golgi, so that these proteins start packing. And there are also some structures called endosomes. You can see early endosome, late endosome. These endosomes are sorting centers for material from outside of the cell or from the Golgi and sending it to lysosomes. Actually what they do is they just pick up all this information from outside, inside all those vesicles, and send them to lysosome. Why they send to lysosome is because a lysosome have hydrolysis acid enzymes which destruct them, so that after destruction they can either go back to the membrane or they can only go again to the Golgi for the further use in the Golgi itself. Next, let us discuss about an important disease called eye cell disease. Eye cell is inclusion cell disease. Actually, it's an inclusion. For example, you can draw something. Here, something is inside. It's an inclusion body. It is formed because of it is inherited lysosomal storage disease, which is autosomal recessive. There is a defect in N-acetyl glucosamine 1 phosphatase. You can see this enzyme. There's defect in this enzyme. As this enzyme is not present, there is failure for Golgi to phosphorylate mannose. So if mannose is not phosphorylated, this will drop. As this is dropped, now it is actually secreted extracellularly, but now as this is dropped, this vesicle will now go and store inside the cell. Especially it will go to the lysosome. So lysosome, as this is leading to accumulation in lysosome will become ineffective, or it will become deficient in digestive enzymes. So there will be buildup of cellular debris inside the lysosome, and that is why it is called inclusion bodies. So this will lead to coarse facial features, gingival hyperplasia, corneal clouding, restricted joint movements, claw hand deformities, kyphoscoliosis, increased plasma levels of lysosomal enzymes. These symptoms are similar to Hurler syndrome, but it is often fatal in childhood condition. Next, we'll discuss about signal recognition particle. Actually, these are cytosolic ribonucleoprotein which traffic polypeptide ribosome complex from the cytosol to the rough endoplasmic reticulum. Actually, these are the proteins which bring the polypeptide ribose complex from the cytoplasm to the rough endoplasmic reticulum. So if they're absent, then that will lead to accumulation of this protein inside the cytosol itself. Next, coming to vesicular trafficking proteins, you can see this is a COP1. COP1 is backward, it will go retrograde. You can see from one part of Golgi to other, or from the Golgi to the endoplasmic reticulum. Whereas COP2, it will make 2 steps further, antrograde, from the rough endoplasmic reticulum to the Golgi, that is COP2. Clathrin, you can see it is present on the trans side of the Golgi to the lysosomes. It will go to the lysosomes or to the plasma membrane or to the endosomes. This clathrin function is seen in LDL receptor activity, especially in the liver. With peroxisome. Peroxisome is used mainly for oxidation of fatty acids, its main function. So it's 2 types of oxidation, beta and alpha oxidation. Beta oxidation is for very long fatty acids. Alpha oxidation is for branched fatty acids. So if this oxidation of fatty acids is not done, then that will lead to this kind of problems. We'll also discuss that. Not only oxidation of fatty acids, peroxisome also do catabolism of some amino acids, ethanol. They can also synthesize bile acids, plasmalogens, which is a important membrane for white matter of brain. So plasmalogen is important membrane for white matter of brain, which is also synthesized by peroxisome. Peroxisome can also generate hydrogen peroxide. In the name itself, you can see peroxisome, peroxide, hydrogen peroxide. And it can also detoxify liver cells. Now coming to the 3 symptoms, one is the Zellweger syndrome, which is actually an autosomal recessive disorder where there is a defect in PEX gene. So peroxisome is not there here. So because of that, this kind of fatty acids may accumulate and lead to hypotonia, scissors, jaundice, craniofacial dysmorphia, hepatomegaly, and early death. Because peroxisome can detoxify liver. Here there is no one to detoxify, so it may lead to hepatomegaly because of accumulation of toxins and also early death. Now coming to refsum disorder, this is also autosomal recessive disorder, but here the problem is with alpha oxidation. So if there is no alpha oxidation, then the branch chains or fatty acids will get accumulated, and especially there is a phytanic acid accumulation, and this will lead to scaly skin, ataxia, cataract, shortening of especially 4th toe, epiphyseal dysplasia. So the treatment is diet and plasmapheresis. We need to give new plasma so that in that we may get the peroxisome and this will reduce. Next, coming to adrenoleukodystrophy. In the name itself, you can see adrenoleuko. Means here the problem is actually in beta oxidation. So very long chain fatty acid will accumulate, especially in the adrenal gland. Leuko means especially in the white matter of brain. So adrenoleukodystrophy. That acid which is accumulating is because of mutation in ABCD1 gene mutation, that is FVLCFA buildup in adrenal and leuko, that is white matter of brain. And this disease can also lead to adrenal gland crisis and sometimes progressive loss of neurological function and death. Next, coming to proteasome. Proteasome is something like a barrel shaped protein which is mainly used to degrade ubiquitin. It degrades the ubiquitin tagged proteins. So why we should degrade ubiquitin is sometimes the ubiquitin protease system is accumulated in the central nervous system and it is causing Parkinson disease. So ubiquitin should be removed, and this function is done by proteasome, which is a barrel shaped protein, and it requires ATP to remove this ubiquitin. Next, coming to cytoskeletal elements. In the name itself, you can see cytoskeletal. Cyto means cell. Cell skeletal system is nothing but cytoskeleton, and here you have many structures which are helpful to support the cell and also for the cell division, organelles, movement, etc. So there are types of filaments like microfilaments, intermediate, microtubules. So first we'll see the examples, then you'll understand the function. Microfilaments are like actin and also microvilli. Actin, it's useful for muscle contraction, microvilli in the intestine for absorption, and also for cytokinesis, actin is useful. Next, coming to intermediate filaments. These are like vimentin, desmin, cytokeratin, lamin, glial, fibril, acidic proteins, neurofilaments. So all these kinds of examples are useful to maintain the cell structure. For example, vimentin is present in mesenchymal tissue, Z disc, also desmin is present in Z disc. Laminin, you can see here, laminin separates the nucleus from cytoplasm. Keratin is a marker for squamous cell carcinoma, especially leiomyosarcoma and CNS tumors. Vimentin is like a marker for them, tumor marker for them. And if liposarcoma, leiomyosarcoma and rhabdomyosarcoma, for these three tumors, desmin is a tumor marker. Next, coming to microtubules. Microtubules are like cilia, flagella, mitotic spindle. So all these are useful for movement of the cell and also for cell division. So if you see the microtubule structure, its outer layer, its cylindrical structure, right? Its outer structure is made up of alpha and beta tubulins, which is a dimer, and it has two GTP molecules which are bound to it. This structure is actually incorporated into flagella, cilia and mitotic spindle. So in flagella, cilia, mitotic spindles also you found the same structure, that is a microtubule structure. You can see one side it is a positive end, one side is a negative end, and the middle part is called protofilament. The positive end is called heterodimer, okay, because it is made up of different kinds of cells. You can see heterodimer. Next, molecular motor proteins. They are useful as a cargo towards opposite end of microtubule, because you can see this is a centrosome. From here, this kinin, dynein, this form. This negative end represents towards the nucleus. So you can see the negative end is antrograde to microtubule, means from the negative end to the positive end, that is a kinesin, which is away from the nucleus, whereas dynein is towards the nucleus, that is from the positive end to the negative end. So it's a retrograde to microtubule. So the mnemonic is ready, attack, because R and D retrograde dynein, attack A and T antrograde kinin. So this is important. Clostridium tetani, herpes simplex virus, poliovirus, rabies, all these use dynein for retrograde transport to neuronal cell body. So all these viruses and toxins use dynein for retrograde. Dynein is for retrograde, kinesin is for antrograde. And actually these microtubules grow from a centrosome near nucleus, and they grow slowly and they can rapidly disassemble. The drugs which acts on microtubules are mebendazole, which is antihelmintic, Griseofluwin, which is antifungal, colchicine is anti-gout, vinca alkaloids are anti-cancer, which inhibit polymerization. Taxanes are also anti-cancer. Now let us discuss about cilia structure. So cilia, actually, according to its location, it have two different structures. The first one is motile cilia, which contains 9 doublets like this, and in a central 2 singlet is present. This is a arrangement of microtubules called as axoneme. And the second one is the basal body, which is below the cell membrane. The structure, it contains triplets, 9 tubule triplets, but no central microtubule in the base, that is below the cell membrane. It doesn't have singlet in the middle, but above the cell membrane it has singlet, one singlet in the middle. Next, some cilia are non-motile. If they are non-motile, then they work as chemical signal sensors, and they also have some role in transduction and cell growth control. If there is some dysgenesis, then it may lead to polycystic kidney disease, mitral valve prolapse, or retinal degeneration. Especially axonemal dynein. You can see here this is a dynein which connects this 9 doublets outside. So they link to peripheral doublets and they cause the bending of cilium. So if this dynein is absent, then the cilium can't bend. And there are also gap junctions which enable coordinated ciliary movement. So actually cilia is smaller and they move mucus and respiratory tract, but flagella are longer and they are helpful for sperm motility. Next, coming to primary ciliary dyskinesia. So this is a problem also called as Cartagener's syndrome. This is autosomal recessive where there is dynein arm defect. So if there is no dynein arm, the cilia can't bend and move. So because of this, there are many developmental abnormalities like situs inversus. You can see actually heart should be on the left side, but because of this disease the heart may be on right side. So this is also called dextrocardia. So situs inversus is seen, and also hearing loss is there because of dysfunctional eustachian tube, and we may be having recurrent infections, sinusitis, ear infections, bronchiectasis due to impaired ciliary clearance of debris. So all the phlegm will be accumulated because of this problem. And there's also infertility because of increased risk of ectopic pregnancy, because cilia is immotile here, and also the spermatozoa may become immotile. So the lab findings are decreased nasal nitric oxide, which is very important. Decreased nasal nitric oxide is used as a screening test. So the triad for this primary ciliary dyskinesia is chronic sinusitis because of absent cilia movement in your nasal septum, bronchiectasis because again cilia is not working in your bronchioles, and situs inversus, and it's a developmental problem. So this is a triad for primary ciliary dyskinesia. Next, sodium potassium pump. We know that sodium potassium pump works by presence of ATP, which brings 3 sodium out of the cell and brings 2 potassium into the cell. You can see 2 potassium are coming into the cell, 3 sodium are going out of the cell with the help of ATP. So there are some drugs like cardiac glycosides, digoxin and digitoxin, which can directly inhibit sodium potassium ATPase. So there's also sometimes indirect inhibition of sodium calcium exchanger that will lead to increase in more calcium inside the cell, leading to more cardiac contractility and thereby inhibiting the sodium potassium ATPase pump.